Hello, dear friends in Brussels. Thank you very much for the kind invitation for me by the European Brain Council to give a talk at the Brain Innovation Day. An inspiring talk I was told to give. So I think this will inspire you. New frontiers in understanding and treating migraine headaches. The story began in the midst of 1970s. Behind me you see the first peptide we discovered. That was VIP, just here. And that was followed by substance P, neuropeptide Y. Almost every year during this period we found a new peptide in the perivascular nerves. Here is a summary of the findings, like 3 different signal substances in the sympathetic nerves, a lot in the parasympathetics, and in the middle you can see the sensory fibers to the intracranial vessels coming from, in principle, the trigeminal ganglion. And you see, of course, substance P, PECAP, nociceptin and CGRP. 1983 CGRP was discovered. Immediately we started to produce a peptide using a peptide synthesizer. We got the peptide, we could verify its potent vasodilator effects. We also used the peptide to create antibodies, so we could do immunohistochemistry, as you can see on this illustration. The nerves were there in 1985, they are there also today. So that's a great reward for a scientist. We also used the antibodies to create a method to measure the peptide in the circulation, radioimmunoassay. At that time I also proposed that CGRP is important for regulation of the cerebral circulation, and most importantly it's involved in migraine. Together with Peter Goadsby we made the method, the RIA method, available for use in patients. And this is a first study where we verified in patients with trigeminal neuralgia that they can release into the jugular venous blood substance P and CGRP. This was not a big shock to the society, but when we came with this finding, namely that in migraine only CGRP was released, not substance P, not NPY or VIP. It was less interesting because everyone was looking at substance P at that time. When we came to a few years later, repeated this study in patients, we had got Suxatriptan as a tool to treat the patients when they came to the clinic. As you can see here, pain disappeared and CGRP normalized. So that was very rewarding to repeat the study. Again, substance P was not altered. As you can see on the other part of this illustration, subsequent work with Peter showed that Suxatriptan or other triptans, they were not only vasoconstrictors, but they could inhibit the release of CGRP. In this case we have illustrated it with the presynaptic receptor. When we have a look on the trigeminal ganglion with immunohistochemistry, we could see, as you see the red dots here, there were small cells that contain CGRP, and they can release CGRP, and there are also in the upper part fibers that contain CGRP. That's the C fibers. The large green neuron in the middle, that is neurons that contain CGRP receptors, and around there are something called satellite glial cells, and we also find receptors for CGRP in these. A big revolution in the understanding came when the CGRP receptor was cloned and identified in detail. Here you can see that it contains 2 elements. One is CLR, calcitonin receptor-like receptor, and RAMP1, both these elements are necessary for a functional receptor. The drugs that have been developed, they are listed, as you can see, the first two, olsegepant and telkegepant, the group is called Gpans, and they can block this gap between RAMP1 and CLR, and thereby CGRP doesn't have effect. There are other Gpans coming into the market as we speak. We were very rewarded when we tested the Gpans in patients during the time 2000 to 2009. They were very good. All trials were positive. They caused pain-free at 2 hours, and also a prolonged effect was seen. The program was halted, but now it is running. Then we have the monoclonal antibodies. They started to work on these monoclonal antibodies in the midst of the 2000s. So like 2005, I first caught a glimpse of them, and then we did a couple of studies 2007. They are large molecules, 1,500 times larger than the little one, as you can see on this illustration, which is the size of a Gpan. Of course, these large monoclonals must be injected, while the Gpans can be taken orally. The antibodies, they are in principle working two ways. One is binding to the receptor, as you can see in this illustration. But that is for one of them, the erenumab. The other three antibodies, they work by binding to CGRP, forming a complex that does not get to the binding sites also. The antibodies that we have, this is just illustrating this, that they are the receptor antibody that is totally human, the other ones are humanized. The difference between the different monoclonal antibodies towards CGRP is due to the different binding sites on the CGRP molecule to the right here on this slide. How are they working? I just pulled out this one. The upper brown part shows patients. Each little bar is a patient, and to the right you can see almost 30 monthly migraine days, so very serious issues. And then the blue ones going down, that is the effect of the drug. So if I summarize, some patients have a fantastic effect, migraine totally disappears. Half of the patients, they have effects, and they are often helped and happy with that. And as you can see, some of these bars, there are no blue bar with them. So there is a fraction of patients who do not respond. So our issues we can discuss further. Where are the antibodies working? And measurements of different drugs, like you see, Suxatriptan and the Gpans, 2 or 3% can pass the blood-brain barrier. When it comes to the antibodies, in essence, they don't pass the barrier at all. And for 2 of them, it has been analyzed in the CNS, and like less than 0.01% can pass, so it's extremely little. Another issue has been discussed at length. Can the barrier open during a migraine attack? And that is, no, it doesn't. We did some studies, and as you can see, the brain, we injected a tracer, and as you can see, the rat turned almost blue. The brain is still pale, there is no blue dye on it, and below is actually the trigeminal ganglion. I pulled it out, and you can see it's blue. That means that this tracer, Evans blue, it passes the barrier freely. But that is an old method, so therefore we set up the so-called permeability surface product, which is a quantitative method. And you can see that the trigeminal ganglion has free access. This shows that it's almost as freely as to the heart or other parts of the body, while all the small other regions noted, that's in essence no passage at all, and that's different brain regions. So what we can see here is in the trigeminal ganglion, that is, shall we say, one possible site. And we have the red here, that is CGRP expression, and the red C fibers. So they can communicate with satellite glial cells, but also with the larger neurons. And to the right there is a blood vessel, so of course they can also have effects there. This is just illustrating here again this communication. But how do they communicate with each other, or do they not? Well, we have done a very interesting observation that from these neurons there are of course fibers, C fibers and A delta fibers. And interestingly, this red line you can see, that is marking the A delta fibers with ramp antibodies. And what we can see in these A delta fibers, there is something called nodes of Ranvier. That's where it's not surrounded by the myelin. And in the midst of that there is an opening, and that is a possibility, which seems to be a fabulous new discovery. And you can see on this picture that there is the red down here on this thing here. You can see these are the C fibers, and this is boutons on the C fibers, and above that is the A delta fibers with the opening here for the nodes of Ranvier. We have made a sketch here easier for you to understand that in these nodes, the C fibers, the CGRP can be released, get to the CGRP receptors here on the A delta fibers. Cyclic AMP is formed, pKa comes here, and then this can then modify the sodium and potassium channels and thereby induce what we hypothesize its sensitization process. The remaining little story we have and currently is working on is this picture, which you know very well. Below is the age of the patients. The upper one, that is women, and the lower one is males. Why this disproportionality between the sexes? Well, of course, it's been discussed hormones. And can, for example, a hormone like estrogen modify the trigeminal pain pathway? Well, our findings now is for the estrogen ER alpha. We can see them here. You can see this is ER alpha, and you can see that it goes over here to the where you see this nuclear expression of ER alpha. That is very important finding, I think. But for the other estrogen receptor, the ER beta, it is shall we say cytoplasmic. So in different ways these two receptors can modify the expression and function of CGRP in the trigeminal system. So when the levels of estrogen in the blood of the females change during their active years, there can be modification of this pain pathway. So there's lots more for us to work on. And in the caveat, of course, these new antibodies and the small molecules Gpans, they may act. I think this nodes of Ranvier is a key region With knowing and understanding this, we can perhaps develop even better medications for men and women. Thank you very much for your attention.